Sporty TV YouTube promo 10 seconds action. Okay, we got the best match so far and for free. And not just the best matches, we've got top analysis and exclusive interviews. And it's all for free.
Good afternoon, viewers at home. This is another opportunity for us to meet once again at Omega Bio-Nexus. This time around, as you see it display on your screen, this is an opportunity again to do what is called do it yourself at home. So today there are going to be only six slides. Out of these 6 slides, you have only four that will be meant for real do it at home. So the first one is going to be the front page, and the second page
This is another opportunity for us to
Meet once again at Omega Bio-Nexus. Omega Bio-Nexus. This time around, I on your screen. It's an opportunity again to do what is called do it yourself at home. So today there are going to be only six slides. Out of these six slides, you have only four that will be meant for real do it at home. So the first one is going to be the follow class at home, and the second phase is another opportunity for us to meet once again at the next one. So already display for you is a sequence search with a blast, the use of NCBI. And don't forget what NCBI or blast, what they normally do is just to compare our nucleotide base, that is the DNA, as well as if you have a protein sequence with what you have in the database. The steps that you are expected to take note of include you have to go and retrieve either DNA or protein sequence. Go and retrieve it on the NCBI. By the time you retrieve that, you are going to do what is actually meant for you to do. That is, you paste it in the NCBI Blast website. By the time you paste it, you are going to select the database. That is, are you going to make use of a NR or NT, which is actually meant for nucleotide sequence? So you are going to run the blast, and you are to document and screenshot each of these at every step. You have to screenshot this at every step, then you'll be able to record it. Don't forget that I said whatever you are going to do today, that is the do it yourself that you are going to do at home, you are expected to start documenting it. You write it somewhere so that for you to be able to submit immediately, the screenshot one you are going to submit not later than 30 minutes after this exercise is already up. So please do that so that we'll be able to submit, and you have to submit the hard copy on Monday. To submit the hard copy on Monday, please do that. So the first exercise you are going to do is to run the blast, which is actually meant for you to do following the instruction at on the screen. Follow that instruction on the screen, so you'll be able to do it. Thank you. So in the next few minutes, I'm going to go to the next slide because of our time. I'm going to go to the next slide because I don't want you to waste time in what you have already. So don't waste time. In the next few minutes, each slide is going to last for minimum of 5 minutes. So we have four slides all together. So when we say minimum of 5 minutes, that is minimum of 20 to 25 minutes, I mean to 30 minutes maximum. That's when you are going to stay here. So and by the time you stay here, immediately after that, I'm going to count another 30 minutes, and I will be expecting you to send the screenshots to me. Send it as an attachment on my website, on my email address, which is actually txy23m@yahoo.com. txy23m@yahoo.com. T for Tunde, X for xylophone, and Y for yogurt. 23m that is 23 Michael at yahoo.com. So you submit that. Not only that, you are expected to also comment under this lecture. You are expected to comment, like, then you are expected to mark the attendance here. Then later on, you are also expected to look at similar work or similar exercise on this Omega Bio Edutainment as well as Omega Bio-Nexus. You can check for details that are related to bioinformatics so that we'll be able to know exactly how to prepare for the mid-semester test. Then don't forget that already you must have seen the adjusted timetable for the mid-semester test. So it is actually shifted forward so as for you to be able to adequately participate in it so far. And don't forget that I told you that I'm going to have 79 people that are coming from microbiology as well as almost 50 people that will be coming from environmental biology for the mid semester test. So please try as much as possible to do this. Okay. So I will be waiting in the next 3 minutes. This is going to be off. This is going to be off in the next few minutes. So before you go to the next exercise, go to the next exercise. Okay, don't forget to go to your browser. You type in EMBL EBI. So thereafter, you now type Cluster Omega. Go to Cluster Omega site. It's going to take you there. And already in the first exercise, you must have gotten your sequences. Don't forget that we normally work with faster formats. So the faster format of the sequences that you have, so you are going to get them. So don't forget that you are going to get more than one sequences. I told you that in the comment section, and I've written it there that you should have more than multiple sequence alignment is for you to be able to compare more than one sequence, more than three sequences at the same time, so that you'll be able to know the similarities as well as the differences. So what you do is you make sure you get like 5 to 10 sequences in their faster formats. Then don't forget to save them in a file or in a folder. So by the time you now, you must have now opened your Cluster Omega site. So it is that folder you are going to upload in form of the, since it is already in faster format, then you click on run. So when you now click on run, you'll be able to view, you'll be able to view the alignment. So it is this alignment that you will now screenshot and you will paste. You screenshot and you paste so as to actually give me your results. Okay, so don't forget. So I want to believe that you must have done that as well. Let's go to the next slide for the next exercise. So that is a basic genome annotation using of Ensembl and UCSC. Okay, so it's either you choose to make use of Ensembl, which is a e-website, or you choose it to be UCSC genome browser. Okay, so what you do is genome browser, this kind of these two tools, bioinformatics tools, will allow you to be able to explore different locations on your sequences or on your genes, sorry. So and the steps to be able to do that effectively and efficiently is already displayed for you. Okay, so what you do is you open Ensembl, then you must have one gene in your mind. Maybe you are interested in a special gene, wanting to know whether that gene is present in a specific microorganism or specific organism. The organism could be plants, it could be animal, it could be prokaryotic organism, it could be eukaryotic organism. So you must have been interested in certain gene. For example, you must be some of us can be interested in the gene that is actually coding for maybe beta lactamase, that is an enzyme that is very, very important for glycolysis. You could be very, very much interested in that. Okay, so we want you to use this to view different features and context in this. So you should be able to do that. And don't forget that I told you that once you are in confusion over anything, so the next thing is you go and consult your chat GPT. So for example, if I were you, the first thing after I must have documented this is to go to chat GPT and prompt chat GPT to tell you how can you make use of Ensembl. How can you make use of Ensembl to search for any gene of interest? So even if you don't know any gene at all, you can even ask the chatbot, ask ChatGPT to tell you list different types of gene in a certain organism. So all those genes you can just pinpoint anyone, just pick anyone or two in that, and you'll be able to paste it in this website, and you'll be able to know what it is actually meant for and how it is actually looking like. Okay, so don't forget that we have less than 10 minutes to go. Less than 10 minutes to go. Don't forget that this exercise is expected to last only for 45 minutes. Thereafter, maybe 30 minutes after, that is when submission is going to be expected to be submitted. Don't forget you have to submit once again at txy. Okay, you are going to see it in my comment section. Okay, txy23m at yahoo.com. Okay, it's already documented there. Send your final results to txy23m at yahoo.com. Okay, that is where that's going to be expected. So don't forget we are live at Omega Bio-Nexus, and the program before you is a virtual run, or what is called a virtual do-it-yourself or home-based kit in bioinformatics. And the expected students for this basic bioinformatics are environmental biology, microbiology, or any biological science field or area or career that is interested in the use of bioinformatics as a tool you want to use in your own career. Don't forget that I told you that now bioinformatics is no more just a course. It is a discipline. It is a career. It is a profession that people are actually making use of, and they are using it in their long career in processing of a lot of information. This is the era of machine learning and artificial intelligence. So bioinformatics is relying solely on machine learning and bioinformatics, machine learning and artificial intelligence to run, analyze, to explain, to explore different types of information in about organism. You want to know, you have gotten a sequence of an organism, and you want to know whether that organism or that sequence is actually related to any organism in the whole world. So what you do, and that's why you run a blast. Then you want to know multiple sequence alignment tools, that is MSA. So that is, you want to know what are the differences and similarities between your gotten sequences and some specific sequences that you must have known. Okay, so um so let me run through again. Don't forget, this is bioinformatics for beginners, and that is a home-based kit, which is a do it yourself. That is what you can do. And as you can see it, you can see that guy holding his smartphone, and is also making use of bioinformatics tools in analyzing the DNA sequences. And in doing that, he is doing that at home. Okay, and just like I said, introduction to bioinformatics and the web tool. All these you don't need to run them, or you don't need your PC, a laptop. It's not necessarily that you must need them before you're able to make use of this type of do it yourself. So bioinformatics is the use of computers. Your computer could be your phone. Your phone computer could be your personal computer, like a laptop and other computers. So what do you use it for? You use it to store, retrieve, and analyze biological data. It is until you use your computer to be able to perform all these tasks before you'll be able to know that you are practicing bioinformatics. Don't forget that in bioinformatics, basically what we make use of and what you normally do is we work with informational molecules. And when we are talking of informational molecules, these are something that involve DNA and RNA and protein sequences in most of the time. So it is this that can actually give us the clue into how these informational molecules is being used. So it is used to identify genes. So our do it yourself today be able to help us to be able to identify genes, okay, compare genomes, compare genome of one organism A with organism B, with organism C and other organism. Then we also make use of bioinformatics in designing of drugs, and that is the bioinformatics that you are going to do in the next time. That is advanced bioinformatics that you make use of it in designing drugs, discovering or recognizing or detecting what is called drug sequences of drugs that have drug potential in them. Then you can also use bioinformatics for vaccine development. You can also use bioinformatics in identifying resistance in a specific organism. You can use it to predict whether they are going to be resistant or not, and which gene is going to be actually being going to be responsible for this. You need it. So, and we are going to make use of web-based tools that you don't need to actually download them. You just, once you have your internet, you'll be able to make use of these tools that we are talking about. That is NCBI. EBL, EMBL, EBI, Ensembl. Then you'll be able to use UC, SC genome browser. So these tools will allow you to be able to perform and analyze directly from your data. It is web-based. It is web-based. It is internet-based. Oh, with no installation that is required. And the first one you are going to do, you already have it, is a sequence search by the use of NCBI, that is a blast. You have it already displayed for you. So the second one is to be able to make use of all these web-based tools for multiple sequence alignment. So, and it is already displayed with you. MSA align 3 or more sequences together to be able to identify conserved regions. If they are conserved, if there are regions of that are conserved, that are common, that means the three organisms must have the same, they must have originated in terms of evolution, they are related. So irrespective of the source of your sequences, once when you use the MSA, you now identify conserved region. That's going to tell you that such organisms or such sequences are from the same evolution, the same origin. Okay, and the last aspect of what you are asked to do is to be able to make use of open Ensembl to be able to identify different region on your genes and location. This is actually helping you to be able to annotate your gene. So, and lastly and conclusively, so here when we use NCBI Blast, that is for sequence search tool that you use, and EBI Clustal Omega, that is for multiple sequence alignment tool, and Ensembl slash UCSC genome browser is for genome exploration and annotation. These are freely accessible tools which